We'll have a break in just about 1 hour, but right now we'll move on to our second keynote speaker of the day, and that's Dr. Jianming Yu of Iowa State University. Dr. Jianming Yu is a Pioneer Distinguished Chair in maize breeding and Associate Professor in the Department of Agronomy at Iowa State University. The focus of Yu's program is to combine maize breeding with cutting-edge genomic technologies to address significant questions in quantitative genetics. Dr. Yu's research integrates knowledge in quantitative genetics, plant breeding, genomics, molecular genetics, and statistics, and has the ultimate goal to develop and implement new strategies and methods in trait dissection for crop improvement. The title of Dr. Yu's presentation today is Genomic Selection, Historical Context, Technical Details, Empirical Findings, and Perspectives. Everyone, please join me in welcoming Dr. Jianming Yu.
Thank you. Okay, yeah, thank you. Great. Thank you, everyone. I somehow give this title to grad student, and that set up like a review article, right? From history to the future. So last night we had a dinner with some of the student organizers. I think Francisco asked me a question. What do you do? So I went to online and I compiled my 56 presentations that in the past 10 years, 12 years together. I did a Wordle and trying to see my presentation titles. And showed up as most people would know me as association, as mapping, right? As genetics, genomic, genome-wide. You see breeding here, right? I'm not a big real breeder. We don't release any line, but we work in the general area of plant breeding. And you'll see sorghum here, and you'll see maize somewhere here. Okay, that's interesting to see. We are what we talked about, right? We are what we present. And I'll give acknowledgement. This project, the main technical part of the project was funded by USDA, DOE, the Joint Plant Feedstock Genomics Program up here. And then we have funding. Previously, I worked at Kansas State University for 6.5 years. Up until January 2013, I moved to Iowa State University. Now the main focus is maize. But today, we'll be talking about some of the work in sorghum as we generated some interesting findings. And then this group is, these are our collaborators. This group are my lab. And then sometimes I feel sorry for them because they're not only have to do something that publish a paper, but they have to, under my pressure, to stay on the cutting edge of thinking, right? We're not in a position to competing with any companies and large programs, but we can compete what we think, right? This outline of my talk will introduce the genomic selection. This topic everyone wants to learn about. But then, interestingly, I think the genomic selection should not be something strange to everyone here. It should be familiar with you. So I'll use the breeder's 10 tenets to try to connect this genomic selection to plant breeding very closely. And then you might show you some examples. I'll be playing some numbers. Graduate student, again, you're not supposed to feel confused, right? Because we're talking about these numbers. You have to have some context, right? Pay attention to these numbers throughout my presentation. And then finally, again, I'll go back to the perspectives, say, what are this area heading to? How do we combine the breeder's knowledge together? In the audience, we see, here we have lots of successful breeders. They can also try to feedback. Grad students can have a discussion with their professors about this topic. Again, in Brand's introduction, he introduced this very nice word called selection, right? What is selection, right? It's either by viability or fertility. In most sense, that's we're talking about natural selection, right? Selection is very powerful, right? As some of the slides Brand showed. But here you can see that you selected from a wild master plant depending on your selection target, what parts of the plant you try to select. You can have different purpose for human consumption, right? And then in plant breeding, we know that selection changes allele frequency and genotype frequency. Those are like classroom terms, right? And then we know that selection happens at different stages of plant breeding. And then these are the plant breeders in the field, right? Every time you have a picture of a plant breeder, you have to in the field, right? You cannot in the lab or in the computer, right? And then very successful plant breeders here, right? Either is selecting a single head to be bagged, to make a single head to be crossed, pollinated. That's a selection process. And then when I try to introduce what is not genomic selection first, right? There's a specific term for genomic selection. So selection based on the observations. In Brand's picture, it's the early Asian farmers, right? Try to domesticate different plants, right? And then recently we played with some gene cloning project, right? It's like selection for the Shadowin, which is the #1 domestication syndrome trait, right? Which is Shadowin or not Shadowin, right? We don't need markers or anything, just select, right? Some of the trait that is highly heritable, and then you don't need to replicate the trial, you just select. And then later on, we know that selection based on replicated trial really speed up the selection gain, right? We have to replicate. We're not simply just based on observations because there's a lot of random errors. That's why in plant breeding, single plant observations, typically for agronomic traits, are not trustable. You have to have a plots, right? You have a plots. And then this is a cotton yield trial in Texas, a big major crop here. We have to do this based on, and then if you actually, after you've done your work on the based on replicated trial, give you an example of the selection based on the blood values, based on linear unbiased prediction. So in this case, sorry, I couldn't find a cow or cattle. Here the ram, here to get the blood value or breeding value of this weaning weight of a ram, not only consider its individuals, but also its dam and sire, its half-sieve, its progenies. Together, by pulling the information you collected from all these relatives and its individual itself, you get a blood value. That's very typical if you learn something from animal scientists, and also in plant science, we're doing blood values. These are not genomic selection, by the way. You can think of as genomic selection, but they're the specific terms. Next, you say what is also not genomic selection is that assume you already identify informative markers, very informative markers for disease resistance, insect resistance, or major QTLs for the soybean cyst nematode resistance. You can essentially conduct your marker cyst selection or marker cyst backcross, right? And then you actually can do that, but that's not genomic selection. That's marker cyst selection in the narrow things. And then here, you can also genotype and select based on sum of estimated genetic values across multiple QTLs. As the previous speaker said, if you find the QTLs, you put them together, you think you can estimate a value for this genotype. Again, sorry, that's not genomic selection. That's a marker assisted selection, right? You put it together. So I'll have some equations for grad students to understand where this Q is the number of declared QTLs. Those are QTL partial threshold. And then you look at the QTL, it's genotypes, right? Homozygous, heterozygous, or recessive dominants together, you get a genotypic value. You based on that value do the selection. Again, that's not genomic selection. We have too many slides of that, right? Now, finally, what is genomic selection? We have different levels of understanding. First, you genotype and select based on the genomic estimated breeding values. Pay attention to this formula here. Now, instead of the Q, I switch to M, right? I'm not saying Q, but it's QTL. I'm saying all the markers, M, right? The Q can be smaller. Let's say 13 QTL, 30 QTL, 40 QTL. Now, the M is a genome marker, right? Together, give you the estimated breeding values for each individual. Your selection or based on genome markers, right? The specific procedures will explain to you, but that's genomic selection. Here, you also can interpret genomic selection as a procedure where genetic merits of the unphenotyped individuals can be predicted. because you already established a model training, which you have a group of individuals where you have genotype and phenotype. You marry them together, you have a good relationship, build it up, and then you have a group of individuals where you only have phenotype, only have genotype. You can have a prediction. So that's what we say in the prediction, or sometimes we call, genomic prediction or genomic prediction. And next, you can do your selection. You can complain that prediction was not perfectly 100% accurate, but what else you can have? Other options. You have no options because you have prediction here, right? So the selection can either based on the breeding values to advance next breeding cycle, or you can use this genome-wide selection as a pre-screening process, which I'll show you next slides. Let's assume we have a GBV value, obtained through our multi-environmental trial on the training set, and then we predicted for the unphenotyped individuals, right? This is what we can obtain. But here on the y-axis is a true breeding value. This is something that non-existent. It's something we can always approach, but we're never going to get it unless we try run this experiment for the whole life, 50 years, right? But let's assume we can do simulations, we can understand there's a true breeding value here. Here you'll see the correlation is about 77, which is not that bad, right? Okay, if you actually, based on the GBV value, you do a selection, you cut off the first 10 individuals to run your breeding program. Obviously, because you know what happened, you actually select some ones that their true breeding value not as good as you wanted them to be, right? If you select the top 10 based on true breeding value, which is impossible to do, you'll see that it's up there, right? You'll select those other individuals. This individual should have been included. But then let's change around. Instead of saying that it's not perfect, what we can do is also to setting another threshold to eliminate, right? So eliminate these individuals on the left of the red line, which you can also eliminate on the true breeding values. So instead of selecting on this side, you can eliminate this side. So this is actually a difference. There's a difference. Selection is not exactly opposite of eliminating, because you either can complain that should I be thinking about how close my correlation coefficient equals 1, or should we be thinking about how different my correlation coefficient different from 0? Because as long as you have a correlation coefficient that allow you to do something, otherwise you have nothing else to do, right? All this setup is possible because genotyping is cheap. Phenotyping is still expensive or even get more expensive, so that you can actually use this strategy to maximize the selection gain by reducing the cost. Again, what is genomic selection, right? If you look at the first publication in genomic selection in plants, here Rex Bernardo designed embedding genomic selection within a pedigree plant breeding, where you start with 2 parents and then F1s. Now you can DH all this, generate the DH population. The DH population can be genotyped, phenotyped, and you establish a prediction equation. Then the progenies, you can genotype, then predict its phenotype. It's a cyclic process. Embedding this genomic selection or macro-assisted recurrent selection. The difference is the two is that here you would want to declare the significant peak above the threshold before you use it in your prediction equation. But here, everything goes in first, right? Don't worry about who is significant and who is not significant. And then here, the first perspective paper about genomic selection, very nice diagram talking about how you're going to generate your populations, how you're going to have the next generations, where here you have a genomic selection procedure. So again, there's some similarities between these two figures if you think hard. It is basically, it's all about having a step to improve plant breeding process by better utilizing resources, either time or cost, right? This genomic selection is not something, sometimes it's not even an extra step. It's just some step you could embed in your breeding program where you can make it more efficient, right? That's why we have to talk about modern era, the breeders, not in 1990s, but now, right? We have a lot of great hopes for the everyone sitting here, especially graduate students, to do things that slightly different from the successful breeders, right, in the past. Okay, there's two technical slides. Sorry about this. And then what is genomic selection, right? If you understand what is a mixed model, where you have fixed fed random effect, on the left side, what we are doing is we try to relate the phenotype observed with the individual breeding values. Here we have G, which is the individual breeding value, and the variance of this one is the additive relationship matrix, sigma square G, by the environmental variance. But then this is actually somewhat not that strange to most of people, because remember my example of the ram, right? If the individuals that are more related to each other in terms of the pedigree or marker profile, calculate the pedigree, is supposed to have higher correlations, higher relationship within their phenotypes. You're relating the genotype and phenotype and the individuals. You're assigning breeding values to each individual here. But then if you turn the whole thing around, now with this, for genomic selection, what we are doing is that we try to assign breeding values for a marker, right? If you take a plant breeding class, right, you know the breeding value, it's where concept is very difficult to grasp. But now we try to assign a breeding value for every markers, right? This is the lines, this is the markers, this is the rows and the columns. We change it around. We try to assign a breeding value of each marker. These two, it turns out, are equivalent models. If you go through the algebra, you can see that if you actually convert your genotypic values after adjusting, standardize this genotypic value, the relationship between the individual's breeding value and the marker breeding value, there's an equation between these two, right? And then if actually in a lot of the genomic selection program calculation behind the scene, behind the package, software package, what happens is actually you calculate this breeding value for individuals first. Even if you set up your model like this, you actually calculate the breeding values, because the breeding value is a number of individuals, right? You have 100 line, but the markers can be 7 million, right? Markers can be 20 million, which is very difficult to calculate. So let's say if you ignore the details of this, what you calculate, you get a breeding value estimate, this mu hat, which for each marker, now you obtain this. This was obtained on the training population, the population where you have genotype and the phenotype. Then what you do is you plug in this new genotype. Another group of individuals, you have genotype because genotyping is cheap and the professors like to pay them, right? And you get the send out for GBS. Or here we have digital genotyping at the campus of A&M, right? You run your digital genotyping, you get this marker profile, you plug in this one, you get this predicted GBU value, which you can think of it as the GBU value on this side. This side, you can think about this the average allele effect. This side, you just plug in, so it's GBU value, right? So again, what is the genomic selection? Here, and then we want to say that after you obtain this marker effect estimate, which we can call it breeding value, then you can calculate its G hat, which is the breeding value for individuals, because breeders, we don't breed markers, right? We breed the line, we breed sorghum, wheat, cotton lines, right? So you actually have this value. Let's say you ran another performance trial, right? Remember the cotton performance trial. You get a new phenotype for that individual. Then you will run a correlation that can be called prediction accuracy. Sometimes we need to adjust that correlation with the square root of heritability, which is reasonable, because what if you just don't do prediction? You just run your trial again and again, you have a repeatability or heritability there, depending on the germplasm you're using. And then you adjust that correlation, you get a prediction accuracy. So this is essentially adjusting your prediction accuracy by the maximum prediction accuracy you can achieve. If you just keep doing your phenotyping, no worries about markers, no worries about genome-wide, all this stuff, right? Okay. And then when students got introduced about genomic selection, they typically hear these terms, G-blob, base A, base B, base C, and kernel-based methods, right? Gaussian kernel and exponential kernel. But then the reason why we have to have these kernels is not because we want to get it too complicated. The reason it is actually because what is distribution of quantitative trait variance, as Michael Gross showed. Some of the traits might be controlled by polygene with very small effect. Some trait controlled by a few major QTLs. So that's exactly whatever went under this, developing this method. The first one is very easy to understand, called GBLOB. Assumes that this marker effect, the marker breeding value, are from a single distribution with zero mean and constant variance. Everybody the same, right? That actually does not interfere with your estimate of that mu hat, the marker breeding value. This is just some assumption for mixed model to run. You have to have this extra component plug into the mixed model equation so that I can obtain the unique solutions. And then here, base A basically assumes that you have different distributions. Not only have distribution for the effect, but also the effect was drawn from another distribution that characterize its variance, right? And then base B basically saying, what the heck, I know that many places you see that the first speaker showed the GWAS plot. In many of this genome chromosome, there's absolutely no signal. Why I have to assign this any distribution? So we set them as zero. You have pay probability or pay proportion of these markers setting as zero. Just go ahead and set it as zero. And then the other ones, if the variance is zero, the mean also zero, the effect also zero. And then the other one, you give a inverted chi-square distribution. And then base C also further change that slightly by setting mu equals zero and then the distribution to be a constant. So we don't have to worry about unless you are really into this detailed program running, but I'm trying to connect you with the quantitative genetics about the trait distribution. And then again, kernel-based method is very powerful, especially when you have the dominance and static effect considered. But in plant breeding, that is less of a concern at this time, right? Okay, we talked about what is genomic selection, what can we do? We know genomic selection has been extensively studied in recent years, and we have studied its prediction accuracy, different models, and for different trait architecture. If you see two publications have different results, you understand maybe because the trait genetic architecture are different. Or maybe one did extensive phenotyping, one did not do good enough phenotyping. Maybe marker density is the same or not the same. So don't get confused there. And then what is nice about genomic selection is that allow you to optimize the resource allocation, as I showed earlier with the with the slides, right? You can think about really plant breeding is a numbers game. In the past, we know this for ages, right? But then with genomic selection, is a really a numbers game, right? Because used to, plant breeding a numbers game, we talk about how much plots, how many acres we're going to plant. But now we say, wait a minute, how many individuals we can genotype first before we decide whatever winners go into the field. So you have extra layer to increase your large inference space, your searching space. Plant breeding is a numbers game. And also, we're talking about genetic gain per unit, time, and cost. We talk about this unit time and cost because genomic selection can be done, like this previous marker selection, can be done under the conditions where you don't have to have a regular summer to get your agronomic traits in the field. You can speed up the process. And then the cost is because the genotyping is keep getting done, which the previous speaker talked about this capillary system and all this next generation sequencing, right? And then genomic selection has been well integrated into many plant breeding programs, particularly private companies. And then we can do inbred development, test cross-evaluation, and then the parental selection before you have generated the breeding process or the hybrid prediction. You see lots of papers on genomic prediction. So, and then there's emerging areas in the research is genomic selection under G by E. This then is something we really don't like to do, right? every public breeding programs, I'm sure we try to avoid this G/E because this thing is totally out of control. That we want it to be dry, it's going to rain like hell. We want to be raining to test the yield potential. We don't have a dry span, a heat wave, right? We don't like it. But, you know, if you want to do the large scale, if you're in from space, which we'll come back to this thing again, you have to study G/E. You can model the G/E prior to genomic selection, or you can do a reaction norm model where you create a variance-covariance matrix for the environment. It's like we create a variance-covariance matrix for the genotype. And then you can actually integrate the genomic selection with the crop modeling. So we haven't spent enough time because we could not get crop modelers to work for us. Now we recently got internal funding. Really, I just basically tell my crop modelers, get this done, and I pay you one summer salary. Okay. So sometimes in breeding companies, they can have everything work together, but in public plant breeding one, we need to actually work together. The funding ties us together. You can use genomic selection for the exotic germplasm integration. We've only seen a few simulation papers. We like to see more empirical studies if you're interested to do. And then here I'll show you an example of genomic selection for germplasm enhancement. And we can actually spin this to a larger context too, right? Okay, now we talk about all this crazy stuff and the equations, models, the symbols. Let's go back to review the breeders' tenets. All the breeders here, Seth, you know, Bill, and then Wenwei and other breeders, cotton breeders, Wayne, and try to take a look at this, right? So at a meeting, NADP meeting last year at the Minneapolis, we have a keynote speaker, Roger Broomer, and here is his picture. He gave a very nice talk called 10 Cents of a Successful Plant Breeder Understands. Remember, you have to understand this to be successful, right? You want to be successful, you understand all this stuff, right? Okay? So I took out a few of them, like I take out like 8 of them that I can relate to what we're doing or the future of genomic selection to this plant breeding, right? I'll show that briefly, you know, best population, uniform plots, heritability, additive variance, marker selection. Here, this one is interesting. This is a successful breeder, retired, and then he says that genome-wide selection requires experimental verification, right? So we're going to run experimental verification and to show that it works because there's publications that you've probably seen, but there's probably you've never heard about this validation, and there's a lot of publications need to be done on this too, right? And then you can say about checks. What about checks, right? And then time cost. We'll address each one of them. And then plant breeders, you can tell me if you have number 11 or number 12. We can try to learn from you and then tie this all together. Okay, now for the real example we're going to be doing is actually the bioenergy crop breeding and genomics. This bioenergy crops is not something we have been studying for ages. And it's actually great because we get to have a new start. We can think about a term we can call the genomic assisted breeding. You can design your breeding program, pre-process from scratch. Now, not only has to repeat what has been done for the maize, the rice, and sorghum, cotton that 50, 60 years ago. You can just start with new. But remember, you have to remember a lot of old wisdoms. You cannot just let marker take over, genomics take over. You have to understand a lot of good things has to learn from the past, right? And then when we're doing this project, we ask 2 major questions. How do we tap into the plant germplasm, right? This is something that's new. And also, we want to ask another question. When we generate this data, how much information content this data contain, right? Can we maximize this, condense or maximize this information within the data? We can extrapolate these findings to a much broader context, right? And then, so we worked on the biomass sorghum. So we want to make sure we start right with available germplasm collection. We worked with the green curators. We select from initial biomass accessions, which we define, remember, base populations, right? We want to start somewhere. There's a 44,000 sorghum accessions in the green. And then there's 78% of them are photopeer sensitive. You can argue there are other sorghums manipulations to be done with for bioenergy, but we thought, well, this is a great starting point. If they're photoperiodicity, which is grew them in the field, they're just accumulating biomass. We just measure them, right? Then we went through all the steps to narrow down our base population from the seed availability, plant height, some of the characteristics, the possible data. We narrowed down to 3206. Then we select 1,000. So here, now under this context, we narrow down our base population from the large circle eventually to 1,000. We'll get to play with, something we can actually have, manipulate, right? And then here comes the numbers again. So we're doing something called the selective phenotyping. This term is different from what you learned selective genotyping. A term called the selective genotyping, that's when the genotyping is very expensive. So we will just phenotype individuals and then pick the tails, do a selective genotyping. Here, they turn that around. We'll go ahead and genotype this 1,000 accessions first. Then based on marker profile, we'll select 300 most representative ones, and then phenotype this 300. So it's a selective phenotyping, right? There's a lot of literature about selective phenotyping to maximize these allele count numbers, gene diversity, recombination breakpoint under these biparental populations. So you not just go ahead and do something. You think first and design a design and then do something. So we speculate that this selective phenotyping also can be increasing your prediction accuracy. If you missed the other slides, I have another slide to show you again. So what we did is actually we did run a GBS, genotyping by sequencing. You can say sequencing for genotyping. You can say digital genotyping, all accessions of 1,000, establish our base population. And then we phenotype the 300. With the genotype phenotype information from the 300, we establish a prediction equation, right? We just solve the equation to get a mu hat for every the markers. Then, because this 700 out of 1,000 already, we have the genotype already, because remember, we genotype in the first at the start. We can predict its GBU values, right? 700. So before we say anything else, let's look at how well did we do. This is cluster tree plus a heat map of genetic relationship. This matrix about 1,000 by 1,000. You see the clusters of germplasm. This is not a surprise for everyone. Every time we run the markers on the breeding germplasm, we see these clusters, right? We see these race classifications, different germplasm. And then this is the 300 that was 1/3, a little bit less, 1/3 out of this thing. So remember, they're not proportionally sampled. They're sampled to maximally capture the structure here, but with a reduced number, then we can run the replicated trial. And then we did the experiment at Manhattan, Kansas, and also Lubbock, Texas. The chromatin company hosted our field testing. A very nice facility there. So I've came to Kansas, Texas for many trips, but not here up there. Kansas, Texas is just so big, right? We're still on the tip, on the upper north part of the Texas. And then, remember, we have the 300, give out prediction, give us the prediction equation. 700, we have the predicted GBU values. Remember whatever Roger Burma said, we need field evaluation, right? We need verification. So we went ahead and selected 200 of them. We planted in the Manhattan again. And we actually planted in Lubbock, Texas, Lubbock too, but that was because of G/E, because of the uncontrollable weather was wiped out by flood. But it's okay. So we have at least one site as a validation of the 200, okay? So here are some numbers again about the prediction accuracy. And then here on the x-axis is the training population size. This typically people will show. As you reducing your training population size from the original set down to smaller set, you might see a decrease of prediction accuracy. And here are the three traits, staggering trait, root logen, a biomass yield, is the prediction accuracy. Because our 300 was purpose selected to be representative, reducing that number does not change much, right? So prediction accuracy stays about flat, about the same. And then this is the model performance. Remember, we talked about base A, base B, base C pi, and the kernel-based methods, right? So you see that they just overlap each other. This concept here is a G-blob2. That's equivalent of G-blob2. As you actually changing the models, doesn't matter, right? The same if you actually have a relatively adequate sample size, all the models will predict the same thing because you maximize information content already. And then remember, we have this 700 predicted. If we actually have x-axis accessions from 1 to 700, the y-axis is the GBU values we sorted. This weird shape is just because this is the 1 to 700 and it's sorted values. And then when we were designing our validation set, we're a little bit conservative. We say to show this is working, maybe we should pick the top 50 predicted values. We should pick the bottom 50. Remember, we talked top 50 and bottom 50. That almost guarantee will be successful, right? But that seems unfair, right? Oh, let's just do another one with a random 100 that both tails got cut off. We select another 100 that distributing between those extremes, try to have a more or less fair evaluation. But then we can combine all this together to see our prediction accuracy. So this 200, remember, we're going to go to the field. We actually have the field evaluation, right? This is what you get. It's great. You can actually look at this picture saying, oh, why it's not perfect? We're very happy with this. Look, as expected, our red dots is the predicted high. The other green dots is the predicted low. Look at here. This group is higher than this group, right? 2 sample t-test. We're great, right? And then look at what happens in a random 100. They're more spread. They're in between. If you ran the correlation coefficient from just a random 100 because Bell's tails were cut off, you get 42%. And then if you ran the whole thing together, you got 67%. All 3 parts together, you get 70%. Again, remember I asked you to think, if you thinking about how this R is away from 1, you're a perfectionist, right? If you're thinking that how my R is different from 0, you're optimistic, right? You're very optimistic. We should be very optimistic, right? So this actually low biomass ones, they don't just grow much. They're just late tillers, greeners. Look, these are strong ones. Don't have to big bulky the stems. That's what we learned from breeders too. You have to adequate tillers, but tiller uniformly, grow high, right? And then let's see the other one, plant height, right? The plant height predicted. Remember, the plant height predicted. This prediction was mainly done by only done by biomass yield. You can still see that high biomass one tends to have high plant height, and then low ones have low plant height. And then 2 prediction accuracy was about the same. And then the tall ones is really tall and then stand up and not lodging, right? So we talk about lodging. Let's see what happens to lodging. So lodging, you see that this is a GBU value, this is the root lodging. Here, the red one switched to the corner. Remember, the red one used to be the high biomass one. They have less root lodgings. That's why they switched to the corner. Remember that, right? So then the green ones has high. They predicted the GBU value to be high lodged ones, and then this also observed lodging is high, right? And then you can see this is standing straight. You have a lot of ventilation going on. There's a very nice environment. If you go in to measure some traits, you have no hard time. But what if you try to measure this very heavy lodged ones? If you talk to our breeders saying that this lodged ones, how do you actually make sure the harvest starts to pick up those biomass? Some of them is lodged early in the season, just rotted, and there's not much biomass left for you to harvest, right? Again, so everything worked beautifully. We say that the biology crops provide a great opportunity for us to do something, design a little bit differently, right? And then we ask a question. We're not done yet. That's what I'm thinking to my grad students. You're not done yet. You have to think, right? So why did the thing work out nicely? We cannot think about, we did all this great stuff. We narrowed down our germplasm. We have a base population. We did all these fancy terms, right? But what really, if you talk to plant breeders, what do you say, right? Plant breeder will tell you this, which is somewhat, there's large amount of additive genetic variance for most quantitative traits. So that if there's large amount of additive quantitative genetic variance, that means you can do predictions. We know that. Because the mixed model, the GBLAP, all this we're handling is additive genetic variance, right? If individuals more related to each other, their phenotype should be also close to each other. And that's what all the prediction is doing, right? We capture the additive genetic variance. So then we go back to the some of the breeders' tenets, base population, right? Now we are switching to the perspectives, right? So breeders tell you that you need, at the start of your job, you need to define and then your base population, not for genotype, but also for environment, right? If you station it here, what is exactly breeding area you're designed to breed? You can't breed for every whole Texas. Texas is just so big, right? And I mean, in Iowa, there are different maturities too, right? In soybean, there are different maturity zones too, right? So then you can ask the question, is it possible to define the base population of genotype at germplasm collection level? This is not for single plant breeding program. This is for our people that were academics who are exploring this possibility, right? You can also ask a question, is it right time to GBS all the accessions of species in the gene bank, right? This was proposed many times again, I'm not one talking about this, right? And then you can think about, are we saying that one can generate the potentially, look at this word, potentially, right? Functional GS model. You can think about this, right? Because we started with 44K to 3K to 1,000, but we can expand our prediction to older circles, right? And then phenotyping. Again, let's review breeders' tenets. Uniform plots strengthening the association between the genotype and phenotype. You have to do a good job in phenotyping, right? All this phenotype work, I was in the field, working closely with every grad student postdoc technicians because I want to get very good phenotypes to make the genomic selection working. And then breeders' tenets too, use heritability of traits, achieve maximum gain per year. We actually calculate, after we collect all data, the first thing any grad student to do is to run the data to calculate heritability. If it's a biological population, if it's a diverse population, we call it repeatability, right? There's formulas for you to have to get just for your phenotype. Leave your marker away. Just look at how robust your data collected, right? And then we can ask question, is it more important to make sure the phenotype data and the training set reliable? What about the validation data set? Remember, we did 3 sites for the training, but we only have 1 validation set, right? That is a potential, could be a potential reason that our prediction accuracy is not as high as we want it to be. If I do more phenotyping my prediction trait, whatever being correlated with my EV value can be more accurate. I might still achieve more, right? Higher. Can we achieve higher accuracy with high throughput phenotyping, as Mike Gore showed? We are trying to think about all this large context, how this high throughput phenotyping can help us increase the prediction accuracy by monitoring all these details, have the data we cannot collect it by hand, right? Will there be many startup companies that are focused on predicting high accuracy phenotypes at the field level phenotypes? If you ever, go to the plant animal genome conference, it's about these vendors, tons of vendors that are selling the genotyping, sequencing, the seeding library, all this stuff. But there's not many people are doing the phenotype, right? I mean, the grad student and faculty, sometimes we see, at least I thought we have limited amount of capacity to run a robust phenotype. We rely heavily on grad students. Remember, we cannot be the one collect everything. We are just as good as how you can do your job, right? In terms of collecting the phenotype, analyzing them, and run the GS model, and run the prediction. That's why we pay you. It's not high enough, but we hope you have fun here. Okay, so we also started working with companies, engineers called Ackpixel or RoboFlight. We're admiring this, octocopter, right? It's a very nice thing. to have. When it's fly over, it takes 2 hours to get it set up, but when it's in the sky, 15 hours, we collect everything, right? It's beautiful. And then look at this, right? It's beautiful. You actually can see, this is our research plot. These are some of this is our research plot too. These are boulders wrapped around. You can only see that. You can get estimate not just the indexes in DVR values, but also you can estimate the height. Look at this, right? This is actually, the height is also segregating, the plant architecture is segregating, but we need to work with the engineers to extract the data so we can analyze. That's what we're doing. You can dissect into the plots. You can calculate the sorghum canopy up-tier classifications, change that to numbers. We want that to be numbers. We don't want to stop at the beautiful picture level, right? It's enjoyable to see this, but we want numbers, we can run it. And then we can randomly compute this repeatability of this imaging data, right? So how about the environment? I showed you this environment, major sorghum growing area and a testing site. So breeders Tenets saying that you include check genotypes in breeding experiment. Now how you will analyze the experiment before you plant it. So what we did not do is actually we did not have any checks. We're actually really lucky. Now I'll explain why, right? Should we rely on genotype variables to generate the kernel function, which is the covariance matrix among different genotypes, in particular training for an validation stage? Or should we have a small subset of training populations embedded within our validation set, right? What if, like the year we're running experiment, weather changed, or the G/E switched, and then our prediction accuracy is bad? We're blaming the equation or blaming the weather? We have no way to see. We can see, we can tell, but we don't know exactly to present the data, right? What if we are embedding a subset of training population that capture the dynamics of G/E pattern nicely, embedding them to the training set? If something happens, we have a reason, right? We have perfect reasons, we have data to show that. So breeder also telling us that defining and exploiting the base population of the environment genotype. So I did not touch anything about G/E, but you can think of this one. If we can squeeze all this multi-dimensional marker data or genomics data into one-dimensional on the x-axis, if I can squeeze all this weather pattern, environment, everything together on the y-axis, environment, and then that's the G/E pattern give you the performance data, right? So the genomic prediction under G/E is really complex. Hopefully for some individual thesis project, you want to get away from this environmental parameters. You just homogeneous environment, have your prediction done, learn how to do this. That's great. And then here, you're saying we can ask questions, are there better algorithms to design a training population? What we did is actually to maximize the genetic diversity from the 1,000 to the 300. You might be able to do something different, which is great, right? And then can we optimize our testing sites? Is your testing sites is a good capture of the environmental landscape where it's your base population of environment, or you just put on somewhere you can get a plots planted, harvested? And then should we consider the design as a progressive context so that validation information can be used updating your prediction models, right? You're not just validate for the sake of validate at that stage. You'll validate for the whole progress. And then are there other options to optimize the validation set? Remember, we picked the top 50, bottom 50, and then random 100, but you could do it differently, right? And then embedding checks, right? I told you. So we have a lot of perspectives, but if you want to answer, you want to agree with me, it's not easy. I never say it's very easy. It's very challenging. But if you say yes to all these questions, so we'll switch this to a positive perspective, right? Because it's challenging. That's why we have you all here. Try to learn, try to do something different, right? And my take-home message, genomic selection is more than a difficult to understand equation. And then genomic selection is a tool or a strategy. And then can optimize the breeding process. It's evolving. You actually can start changing your QTL mapping experiment to genomic selection just overnight, if you want to think about this, right? And my math slides for the grad students sitting on those tables, if you want at the social hours, you challenge each other, ask a question. What is genomic selection? Thank you.
All right, thank you very much, Dr. Yu, for that excellent presentation. We have time for some questions now for Dr. Yu. We could take 6 or 7 probably from the audience. So if you have a question, raise your hand. One of our wireless microphone assistants in the back of the room will come to you. And also webinar attendees, if you have any questions as well, you can definitely type those into the webinar chat room.
Yes. Yeah.
Could you just go over a little bit more about your idea of incorporating the genomic selection models into the crop models? You kind of mentioned it, but how would you actually do that? Can you give us an example?
Okay, thank you. Thank you for asking the question. So I actually did a presentation on the G/E at the ASA meeting, and there we talk about the G/E and we talk about genomic selection. And then on my slide, I also showed that some other people doing this reaction norm models where you compute a variance covariance for the environment. And we showed that what we wanted to do is combine the genomic selection for the crop modeling. So here's what is happening. Typically, when we do genomic selection, we want to predict the final yield, plant height, biomass component, or login, right? That's the agronomic traits we have. But this idea of combining genomic selection with G/E is not, right? Because G/E is so complex. It turns out that a lot of crop physiologists have been doing this crop modeling for ages, right? They have this differential equations, predictions. They do a good job. The capacity of that crop modeling has improved so that they can insert cultivar parameters so that instead of manipulating this final yield, you can predict, let's say you have a crop stage parameters. You don't want to go to the final yield prediction. You can predict the curve parameters for each genotype. So you almost are thinking you can actually, so crop modeling, you can actually obtain the growth parameters or reaction parameters for each genotype. Then find all those parameters into the crop modeling to generate the final phenotype. So instead of just regularly, because remember, when we do this regular plant breeding, we have the final harvesting yield and plant height measurement. But if you have this stage measurement, high phenotyping monitoring, right? You can actually, instead of the final yield, you can predict the growth parameters, curves, changing, turning point, have those set of parameters using genomic prediction to predict those parameters. Then use crop modeling to give you the final prediction of phenotype. But the issue there is that we really have to work with crop modelers. And I'm thinking that I'll be sitting in a workshop to learn this crop modeling stuff. And then if it's not working, we'll try to play with them too. Thank you. Okay, there's that.
Okay, my question, it's a really nice idea and I really would like to employ this, but I work with a crop that, I work with roses. It can be diploid, triploid, or tetraploid, and we can separate that out. But okay, for the first question is, how complicated does it get when you increase to, let's say, a tetraploid level? And then beyond that, the next one would be, okay, well, the other aspect of this is they're all interspecifics, and so you have, 5 or 6 different species involved in several generations past many times. But so it's a really complex sort of multi-species cultivated crop we're dealing with. Mm-hmm. How complicated does it get?
Yeah, I got asked this question a lot of times, and my joke is that if we have funding, we'll figure it out. It's all about how you spend time and the quality time. It's not like just take a look and be gone. You have to quality time. But I think the major challenge, first step, is actually to find this this genome specific or chromosome genome specific or chromosome specific markers, right? If it's tetraploid, diploid, we know it's easy, right? I mean, talk about wheat, right? ABD genomes. But if you can have the markers that specifically is tagging your A genome, B genome, or D genome, that is actually okay. And then remember again, also, if you just have the markers that are set up can differentiate. So we don't want these markers that you don't know where they come from, right? It has to be chromosome, like roses are genome sequenced, or in a process, right? You can have these chromosome, you can specific marker positions. And then remember, if you have phenotype and genotype, all we're doing is to relate genotype, phenotype, generate the marker breeding values. I think, again, if you're positive, you think it's going to be challenging, but it's doable. And then it'll be done, actually. Yeah. Thank you.
We have time for one more question.
So I have a question. How many years of data do you include to get into a prediction? Like, for example, in building a prediction model, you include a lot of phenotypic data to come into that prediction, right? Like,
so are you asking the marker data?
No,
the phenotype?
Phenotypic data.
Okay, so for grad students to practice, because I mean, high-level publications, you read out hundreds of thousands, but if you have a biparental populations, you can start playing. The number could be as low as 150, 200, 300. It's fine. You can always do predictions. What you will not get is a robust prediction accuracy. But let's say you have a marker. We have shown that for the biparental population for regular crops with 10, 20 chromosomes, if you have 100 markers, 200 markers, you can do the prediction. As long as you can do the QTL mapping, you can do the prediction. It's a good practice. It's another way of, instead of finding the peaks, now you try to predict each genotype's GB values. So the number is not a major concern at this time. Let's say the lines you have 150, 200, you can do that. If the number of markers is 100, 200 for the biparental populations, it's good to go. If it's a diverse populations, it's about let's say 1,000. That's good enough. Remember, I did not say that we worked on the GWAS extensively. I have a workshop at PEG on GWAS and GS. Genomic selection has relatively less requirement for the marker densities because it relies on the whole genome prediction instead of finding that peaks. Yeah. Thank you.
We had a couple of questions from our webinar attendees. One of them wanted to know what are the most challenging points to predicting the performance of hybrids.
What are the most challenging questions or the steps to prediction the hybrids? So we also, there are some publications in the literature, and we also running this ourselves. So what it turns out is actually nothing new to everybody. If you learn a good quantitative genetics, you understand. First of all, if let's say the prediction set or validation set has a genetic relationship close to your training set, by default, you're going to have a good prediction, right? You do not want to use a totally different sets of hybrids to predict another totally different hybrids. If the genetic relationship, remember we talked about the additive genetic relationship, right? It'll be good. And also, if you want to make the hybrid prediction accuracy to be high, it'd be nice if at least you have one of the inbred for the hybrid. You have the one of the inbred from the prediction set was also involved in other hybrid combinations of the training set. That's just a specific example of what I just said, right? You have in the training set, you involve A line, B line, C line in some hybrid performance testing. You generate your prediction equation. And then in this validation set, any hybrid with A line, B line, C line will have good predictions. But if none of these parents in validation set was showing up in the training set, then you have not good prediction. Again, it all depends on the setup of the experiment, which we try to challenge to people to show, right? You have to have some thinking before you do it. Thank you.
And the last question that we're going to have for this session right now is from another one of our webinar attendees who asks, as wheat breeding is a pipeline and continuous process, what is your comments on updating training model lines if a breeder wants to incorporate genomic selection into a breeding program in regards to the training model line size and frequency?
Wow, that's a very tough question because now when we do this project, we don't have to worry the daily routines of plant breeders, right? They have their own headaches, right? How to maintain the. I think the key message that they have to have somebody dedicated to serve and work together with the plant breeders to updating those equations. You cannot simply rely on a grad student. Take 3 years to learn what is going on. The next 2 years they're gone, right? I think the critical question, is I have the personnel to do this updating the models and then to sift through all the phenotypes, to calculate the G/E patterns of your phenotypes, and to calculate whether you have enough marker coverage adequacies first before you run prediction. If you don't do enough careful work, it's not going to work. The prediction is going to be bad. But not because genomic selection is not working, because you didn't do a good job of processing all this. That's exactly the reason I want to show this breeder's tenets, right? The breeder can have a lot of input into this whole process. Thank you.
All right. Thank you, Dr.